Hello everyone, this is Bosheng Li from Peking University Institute of Advanced Agriculture and Sciences. Today I would like to introduce our work using the ultra-long reads and adaptive sampling for the plant genome assembly. First, as an overview, by reviewing the plant T2T genome published in last year, it seems the length of the nanopore reads is important, especially what we call this ultra-long reads, which defines the N50 read length larger than 100 kilobase, is quite essential for sampling a larger genome, uh, like in gigabase size. And today in this talk, I'd like to introduce our improvement on DNA extraction method, which enable us to extract DNA fragments larger than 500 kilobase. And we also optimized the library preparation and finally got a good output for the ultra-long reads. And using this ultra-long reads, we assembled plant T2T genome, and occasionally there are gaps. And in this case, we applied the adaptive sampling, which get enrichment on the reads surrounding the gap region, and finally reach our goal that is complete telomerase to telomerase genome. The first is the DNA extraction. The key for our protocol is track the high quality nucleus, because we believe the nucleus can either enrichment and also protect the long fragments of DNA. And later, by gently filter lysis, separation, and precipitation, we got the long fragments DNA. And we test the tissue we should use, and it seems that the younger the better. In the case of the weight, we actually use auto grows in ductlets for 5 days and 7 days, and also the young leaves. It seems the younger leaves can get the younger tissue can give a good quality on the nucleus and also get DNA fragments more larger than 500 kilobase. And by sequencing more than 200 libraries, it proves that the younger tissue get a longer N50 read length. Using the same protocol, we also extract the high-quality long DNA from pepper, tomato, cucumber, and lettuce. And we also optimize the library preparation. Right now, from the Oxford nanopore technology, there are two cases for the library preparation. Either use the TN5 transportase or the ligation reaction to add adapters. We optimize several steps, including the input, the DNA, the reaction time, and other like the washing steps to finally get a high quality long DNA library without short fragments. And for the ultra-long reads outputs, using the above optimization on the protocols, we get similar output compared to a previous study in the weight, but much longer in the N50 length. And in the case for the single cell, for the one specific cell, we reach the maximum N50 length to 130 kilobase. You can see there are more than 5 gigabase of the reads is purely larger than 100 kilobase. Even couple gigabase, the reads reach to more than 200 kilobase. So in the case of chili pepper, we get good outputs, which reach to 40 gigabase per cell. And using 4 cells, we get like 180 gigabase of ultra-long, most of them actually ultra-long reads. So using this ultra-long reads we sequenced, which enable us to assemble, combine with other sequencing technologies, we can assemble a complete T2T genome of the hexaplot weight, which is 17 gigabase in genome size, and also complete chili pepper genome, which in 3 gigabase of the genome size. We also sequence the other plants like the tobacco, the tomato, and the popular trees. And in most of the cases, we have a good proportion of ultra-long reads, and you can see most of them are larger than 60%. And in the case of pepper and wheat, we get the proportion to more than 90% of ultra-long reads of the total outputs. For the maximum read length, in the lettuce, we get the maximum of 5 megabase for a single read, which is longer, longer than what we can find from a human study. So filling the gaps. So even we apply more than 100 fold of coverage, some place of the genome has no coverage. So in this case, we use the ability of real-time sequencing and real-time mapping of the Oxford nanopore technology devices, then enable us to enrich the reads surrounding the gaps and finally can fill in the gaps. So in the case of Arabidopsis, the most complete genome assembly is the Peking University Columbia assembly, this version. It has 6 gaps has been left on chromatin 2 and 4, and we use adaptive sampling compared to the standard sequencing. We get high rearrangement on each gaps. So mapping, you can see most of the reads were mapping to the gap region, which is located on the telomere region of chromatin 2 and 4. And there are also some off-target reads. But compare the coverage, the target region is much higher, significantly higher than the non-target region, and also the read length. Especially for the reads which cover each end of the gaps compared to the partially covered reads, the reads covers each end is much longer. That indicates not only the adaptive sampling, but also the ultra-long, the read length is necessary and essential for filling the gaps. And finally, we get all the 6 left gap of the Arabidopsis field. And by multi-read supported, we actually deleted the 2 gaps. Actually, there are no gaps there, but we fill the left 4 gaps with different size of reads we got. In another case, when we assembling the T2T genome of a watermelon, there's only 1 gap left on the upper telomere region in chromatin 8. By using the adaptive sampling, we get a good coverage on the surrounding the gap region, also the read length. And finally, we can get more than almost 6,000 of specific telomere repeats and finally fill these gaps. And finally, I would like to thank our nanopore small group, which Dr. Dongdong Lu in charge the small group, and Rui Yan and Yongqi responsible for the analysis, Shanshan, Caijun, and Yanxia, they did the bench work. And also thank all the 3 companies cooperate with us for the discussion and also for lending the sequencing devices occasionally. Thank you.